it's been an amazing day for me. The potential of diagnostics in the future, the transformational impact in the way we deliver healthcare is palpable throughout the day. And so delighted to be here. I'm really very grateful to JC, but also the leadership of Danaher for inviting me, a surgeon. Who would invite a surgeon to a diagnostic evening? So to speak today about the subject, which I think is critical importance to me, but also to the health security. It's just 5 years, in just 5 years' time, we will be celebrating the centenary of discovery of penicillin, and that's in 1928, as we all know. But what sort of celebration it's going to be? As many of you will know, untreatable infections, we heard some of that earlier, for which there is no effective antibiotics, are rising fast and already claiming over 1 million lives a year globally. By 2050, drug-resistant disease could cause more than 10 million deaths worldwide annually. 10 times the mortality today, and more than all those from cancer. The figures from the 2019 UN report are a stark reminder of the bleak future we face if we fail to curb the silent pandemic of antimicrobial resistance. And I was delighted to be invited here, considering the amount of work that Danaher is doing in this field. Just to let you know, next year the UN will reconvene on AMR, and the focus again is expected to fall on the diminishing pipeline of new therapeutics. Disappointingly, little progress has been made since the last UN report. But I believe the real challenge lies elsewhere, in our failure to make better use of high-value point-of-care diagnostics. That is why I have titled this talk, Too Little, Too Late. Before I go on to address this issue, let me answer a question that I'm often asked. Why I am, as a surgeon, speaking about antimicrobial resistance? Well, I was trained to cut out infected tissues during an operation and not to think further about it. It is history that drives my interest. And it is my work as an academic in convergent science that has laid the ground for the interdisciplinary approach necessary to address the complexities of antimicrobial resistance. I've spent much of my career at St Mary's Hospital in London, which is in a district called Paddington. My office is a short walk from the very lab where Sir Alexander Fleming in 1928 left his famous Petri dishes to go on holidays, and where the fungus Penicillium rubens grew, a discovery since heralded as the single greatest victory ever achieved over disease. My ambition is to extend that legacy by delivering a purpose-built world-class specialist facility, the Fleming Center, on the same site where Sir Alexander Fleming made his discovery, devoted to tackling the growing spectra of resistance. Bit more of that later. While it was Howard Florey, as some of you may know, and Ernest Chain, who escaped Germany, by the way, just before the war, at Oxford, who realized the significance of Fleming's discovery. It would take a collaboration with colleagues in the United States, believe it or not, including the Department of Agriculture and Rockefeller himself, to translate his research. In 1945, the pharmaceutical firm Merck and the director at the Northern Research Laboratory, Andrew Jackson Moyer, filed patents for the mass production of penicillin, kickstarting the modern antibiotic era. Mass production, however, also poured gasoline on the AMR fire. Overuse is the principal reason for the development of resistance. We have become over-reliant on antimicrobials, not only in the treatment of infections, but also in prophylaxis of surgery, dialysis, and cancer treatment. and in other fields such as agriculture. Fleming himself in 1928, 1930s, foresaw the threat. He said, "The thoughtless person playing with penicillin treatment is morally responsible for the death of a man who succumbs to infection with penicillin-resistant organisms." Our dependence on antimicrobials is so absolute that our only option is to strive to develop new therapeutics at a rate that outpaces the ability of pathogens to develop resistance. We are like horse riders who tries to outrun the express train. We need to be smarter. The vast majority of prescriptions for antimicrobials are issued by primary care. They account for more than 80% of the prescriptions in the United Kingdom, with respiratory infections being the most common indication. The primary diagnostic question in these cases is simple. Is this viral or bacterial infection? More than 85% of the time, the cause is a virus, for which antibiotics will be futile. Yet doctors do not have the means to determine the cause. They are operating in an information vacuum. Existing tools for differentiating bacterial and viral infections, such as the Centaur criteria, are wholly inadequate to the task. They have a sensitivity not much better than a coin toss. The results is that despite the low likelihood of a respiratory infection having a bacterial cause, 42% of patients presenting to their GPs will be prescribed an antibiotic in the United Kingdom. Worse still, in some parts of the world, they will be able to get them without a prescription. This goes against a fundamental principle of patient safety, that we provide patients with the right drug in the right dose at the right time via the right route. Our antimicrobial prescription practices consistently fail to meet this standard. What is the first rule of war? Know your enemy. A key weapon in this battle is the rapid molecular test, which we've heard about today. This can give frontline clinicians the ability to know what they are treating within 30 minutes, improve patient outcome, and help target infections control processes. And by the way, this is not just what's happening in the United States and what's happening in Europe. This is global. Antimicrobial resistance has no boundaries. So precision medicine has the potential to transform infectious disease management even more than cancer care. Infectious diseases arises through the complex interplay of pathogens, the microbiome, the host immune response, and the environmental factors, as we heard earlier. Each of these yields data upon which therapy can be refined. Yet historically, optimizing antimicrobial therapy has been restricted to a single data source, called the culture. And this audience will know better than most. This is a labor-intensive process with prohibitively long turnaround times, and all too frequently, the information provided is of little value by the time it becomes available to a clinician. There's another way. We've heard about it all day today. Omics-based personalized antimicrobial therapy has already significantly improved the laboratory workflows, as we've seen. AI, image processing, and mass spectroscopy reading has halved the turnaround times, while direct from sample assessment will soon allow us to avoid culture altogether. Genomic assessments direct from samples also holds a great promise. We are on the cusp of a paradigm shift, and this new approaches are moved from laboratory to bedside, empowering clinicians to safely use alternative to antimicrobials while educating patients on their risks. Where pathogens are identified, treatment will be personalized at the time of diagnosis, eliminating the need for broad-spectrum therapy. But this is not all. These advances will also allow us to look beyond pathogen. Improved diagnostics, as we heard again earlier, will enhance our understanding of the immune response or immune age and how it can be leveraged. During COVID-19 pandemic, our most successful therapies, dexamethasone and vaccinations, did exactly that. Bringing diagnostics to the chair side will similarly transform microbial surveillance. Findings will continuously uploaded, providing real-time assessment of community spread, transmission patterns, helping to isolate carriers faster, which is critical to prevent a serious outbreak. There's even a bigger prize here, the democratization of diagnostics. For too long, diagnosis has been dependent on health services, not to the patient. Decentralized testing is crucial to ensure patients can access diagnostics information they need to make informed decisions about their health. As we saw during COVID-19 pandemic, rapid point-of-care tests were key weapons in the drive to contain the spread. But this is not all. Only through global, equitable access to diagnostics can we hope to reduce worldwide health inequalities. We have already seen this in action. The widespread rollout of antiretroviral therapy for HIV/AIDS is often cited as one of the greatest healthcare achievements in history. Less frequently mentioned is how point-of-care testing transformed delivery of HIV care, enabling patients to halt transmission of infection and preempt the onset of AIDS by initiating antiretroviral drugs. It's always the orphan when it comes to the diagnostics. AMR is a global challenge today as HIV was 40 years ago. And just as HIV became a global responsibility, so too must be antimicrobial resistance. The development and delivery of diagnostics for all countries is the only way to avoid AMR becoming a catastrophic for all countries. To achieve this, we need equitable funding. In the UK, modelling has shown that widespread use of point-of-care testing for respiratory infections alone could save over $200 million a year. Yet today, in vitro diagnostics, which influences over 70% of medical decisions, account for less than 1% of healthcare spending in Europe. High-performance diagnostics are still undervalued by healthcare systems, and the question is why? I believe the answer comes from down to lack of clarity between budget holders and manufacturers about value. Clinical pathways are complex. Determining the impact of a single diagnostic test is difficult. And diagnostics may also have a broader population-based effect. Yet budget holders only have a partial picture of these benefits. And their difficulty is exacerbated by budgetary constraints. So manufacturers, meanwhile, are for contending with increasing regulatory barriers such as the EU medical devices legislation, which may raise cost and may stifle innovation. The policy goal must be to provide proper oversight for budget holders, but without deterring manufacturers from entering the market. To achieve this, we require new payment mechanisms involving novel ways of incentivizing manufacturers and standard processes of generating value-based assessments. We've heard part of that earlier. An example is the subscription-based model for new antimicrobials, through which pharmaceutical companies receive a flat annual fee, delinked from volume and based instead on the product's value to the whole health system. Procurement could also be aggregated, allowing high implementation cost to be spread over a wider budget. If this sounds too good to be true, it probably is. But it doesn't have to be. if we can balance the needs of the public, clinicians, commissioners, and industry, then the real question is, how do we do this? My answer to this, first to you, is to get them all in the same room. Tackling AMR requires broad-based response across a wide range of scientific and clinical disciplines. But even world-class science cannot address the AMR crisis alone. Science must work hand in hand with policy, and both require genuine public engagement. We at the Institute of Global Health Innovation at Imperial, where I'm a co-director, are launching an initiative that will bring the key players under one roof. This is the Fleming Center. Sorry, you playing the video, guys?
In 1928, Alexander Fleming, in this lab, by chance, discovered penicillin. Alexander Fleming also warned us of the dangers of antimicrobial resistance.
Antimicrobial resistance is a global crisis.
The problem of antimicrobial resistance has reached alarming levels.
Extensively drug resistance to tuberculosis has been identified in 105 countries.
This is Armageddon. This is the end of our current regime of antibiotics.
If we don't do something about antimicrobial resistance, in another 35 years, there could be 10 million people a year dying. The policymakers only move when the tsunami is there.
It's important to not walk away from the battlefield.
We need new antimicrobials. We really need to move now, or I predict that we will wake up too late unless we really up our game.
If we truly lost all our antibiotics, it would be a bomb blowing up our healthcare systems around the world.
To protect our antibiotics, we do need science, but we also need people. We've got to, all of us, change. We really need to change our behaviours.
There is a danger that the microbes will be educated to resist penicillin.
There are multiple factors driving antimicrobial resistance, and we need to tackle these with every direction. This is all about impact. Doing it on an individual basis, you're not going to see the impact that warrants to address this crisis. We are taking a very radical new approach in addressing this global challenge of antimicrobial resistance. The Fleming Center is unique. We will bring in both the public and the patient, because they are the main drivers for change, policy and science, all together in a single building. We need to engage globally with citizens and to change in behaviors in terms of the use and the misuse of antibiotics. The Fleming Center is the first center that sandwiches science, engagement with policy changes. By implementing this approach, I envisage a world in which we will continue to use antibiotics safely and effectively.
I titled this talk, Too Little, Too Late. Please note the question mark. I don't think it's too late yet, otherwise I would have not been involved, at least leading a state-of-the-art building around antimicrobial resistance. But we do need to act now on multiple fronts, One of these is to foster a diagnostic ecosystem which enables our physicians to provide targeted care. To achieve this, we need accelerated development of high-performance diagnostics across healthcare and provide the right regulatory climate for them to translate into practice. Our patients' future and the health security of our planet depends on it. Thank you.